or anything, then please let me know. So I hope my slides are being shared with you. Okay, so now let's begin. So this is your second week with me, and we'll be doing the same thing as we did in the previous session. Firstly, we'll be looking into the week 2 topics, and then we'll be practicing few MCQs, and along with these topics we will also be going through the current literature. I'll also take your doubts if you have any regarding the assignments or any doubt regarding the week 2 content. So in week 2 you have gone through epigenome, ChIP-Seq, DNase Seq, high-C, bisulfite sequencing. So you have gone through all these methods, how to study epigenome, and then you have also looked into the TCGA and cBioPortal and why they are used and how they are used. Then you have also looked into SNPs and copy number alterations. Then along with this you have studied genotype, gene expression and phenotype, and how these all are linked and what are the different methods to study these things and their interactions. Then genome-wide association studies and QTL studies. So we will look into them one by one. So let's begin. So I hope everything is clear, right? Are you able to see my slides? Just give me one confirmation. Okay, thank you so much. Okay, so I'll begin with the previous concept. We were discussing about the proteogenomics. So I came across this review which will be very helpful for you. So in which it is clearly mentioned that why we need to integrate both genomics and proteomics. So the need for integration is that by integrating the genomic data, transcriptomic and proteomic data, this all combines to form proteogenomics. So now with the help of proteogenomics you can analyze variant peptides, because in proteome you will be getting the peptides. So now those peptides are the smaller fragments of the protein, and there are certain peptides which are common among different proteins. So to identify that to which protein that peptide belong, we must have the information of the transcriptome. So because from transcriptome we will be able to know that it will form that particular protein. So variant peptides can be identified with the help of proteogenomics, and there could be certain peptides which are not present in our proteome datasets. So in that case also this transcriptome analysis can also help us, because we are designing the customized protein dataset by looking at our RNA data. So in that way we can predict that this novel peptide might have come from this particular transcript, if that particular transcript codes for the protein which is not available currently in the reference dataset. So novel genes can also be identified, novel exons can also be identified with the help of proteogenomics, as we have seen in the previous session. Then we can also look into the novel splice junctions, pseudogene resurrections. So I hope you know about the pseudogenes. So pseudogenes are also known as kill genes or inactive genes, we can say. So pseudogenes are mostly, these are the copies of the functional genes. So in our body we already have a functional gene, and if its copy is present, then that copy is mostly silenced or inactivated, because to maintain the dosage compensation. So in that cases that gene act as a pseudogene which remains inactive, which do not express itself. But in certain scenarios, either due to mutations or some other phenomena, that pseudogene can get activated. So in that case, to track that gene we can use the proteogenomics, because we have both the transcriptomic and proteomic data with us, and we can integrate them. So in that way pseudogene resurrections can be done, and we can also do gene model corrections, as this was also discussed in the previous session, where if certain novel exon is present and which was not previously discovered, but now due to transcriptomics data we have identified a new transcript which has that exon which was not previously reported. So in that case we will be able to annotate that exon in the gene model. So in that way we can correct the gene models with the help of proteogenomics. So now the question began that why proteogenomics came into picture, because previously we also discussed that there are limited number of genes in our body in humans. For example, 20 to 25,000 genes have been reported, but if we look at the proteins, then there are about 1 lakh of proteins, more than 1 lakh even. So how this diversity comes from? Because a single gene can code for multiple proteins, and it can also code for multiple transcripts, and those transcripts give rise to multiple proteins. But not all transcripts code for proteins, because we see in the transcriptome dataset, for example, if you look into the RNA-seq experiments, you will find there are many transcripts which have been reported for a single gene. But if you look for the proteins, then they were not many. So there is kind of a difference between the transcriptome which is expressed and protein which is expressed. So in this figure you can see that To correlate between the transcriptome space and proteome space, there are studies which have been done, because from proteome space, the proteome space data set is taken from mass spec studies, which are the experimental studies, and the transcriptome data is taken from RNA-seq studies, which tells you which RNA is present in the cell. So now what are the factors which influence this correlation between them, that we see multiple number of transcripts but we do not see as many number of proteins? So the reason behind is the mRNA degradation rate. So in certain cases what can happen is that a gene can encode multiple mRNAs, but some of them might get degraded. That means it does not get converted to protein, hence we do not find that protein in the cell. The second reason could be ribosome binding sites. So as we all know that from mRNA to protein conversion we require the ribosomes and tRNAs. So ribosome initially binds the mRNA and then it calls the tRNA, and ultimately it leads to protein synthesis. So in certain cases what can happen is that ribosome is not able to bind properly to a particular mRNA, and hence that mRNA cannot make protein. So in that case also there will be difference between the transcript and protein expression. Now the third thing is ribosome density. So in cell there are multiple ribosomes, and even in a single transcript multiple ribosomes can bind, which is known as a polysome, when a single mRNA is binded by multiple ribosomes. So in the scenarios where the multiple ribosomes bind to mRNA, the protein synthesis will be faster. But in other scenarios where ribosome is not that dense, it's lesser in the cell, then the protein synthesis will also be slowed down, or either some mRNAs won't be able to express due to the lesser number of ribosomes that are present in the cell. So this could also be one of the reason. Then codon usage bias. So as we all know that in the codon table there are certain amino acids which are coded by multiple codons. For example, if we look at serine, then it can be coded by 4 or 5 different codons. So in that scenario there is one codon which is biased, which is mostly seen in multiple cases, and other codons are not that much observed. So in that case, if that particular codon is not present, so tRNA won't be able to bring that serine to that particular position. So in that case also, in certain mRNAs where that particular codon is not present, so translation will be slower, or it will not be able to get completed, due to which we will see a difference in the transcript and protein expression. Then protein turnover. So protein turnover means what is the rate of synthesis of protein from mRNA. So it could also vary. Then PTM variants, which are the post-transcriptional variants, that could also be one of the reason. For example, if there is methylation or miRNA inactivation of particular mRNA, degradation, so in that cases also this particular mRNA won't be able to convert to a protein. Then peptide sharing among isoforms. So there are certain peptides which are shared among different isoforms. So in that case, when you do mass spectrometry, you won't be able to identify all the transcripts, or you cannot be able to distinguish between two proteins if they share some of the peptide in between them. So that is also one of the reason that although we got the peptide from the other transcript product, but we are not able to distinguish. So in that case also we'll only report fewer number of proteins. Then low abundant proteins. So there are certain proteins which are very less, and with the mass spec we are not able to capture them. So that could also be one of the reason. And then experimental noise. So we all know that there are certain experiments which might suffer due to certain noises or disturbances. In that way also we will not be able to capture all the proteins. So these are few of the reasons that why we do not see as much proteins as much of the transcripts. So these are the factors which influence this correlation. That's why we need proteogenomics to correlate between both of them. And in this paper only they have shown you how to design the experiment and how to analyze the proteogenomic data. So in this they have told that in case of experimental design, if we talk about the transcriptome data, so we can obtain it from RNA-seq studies or microarray studies, and the proteome data can be obtained from the mass spectrometry. So there are different types of mass spectrometry: label-free, ITRAQ and select. So these are kinds of the labeling. If you have multiple samples, so you need some labeling, either isobaric labeling or some other kinds of labeling such as isotope level labeling. So you don't have to go into the detail of this. And then ribosome profiling can also be done to identify the actively translating transcripts. So ribosome will bind to mRNA and then it will translate that mRNA to protein. So if ribosome is binding, it confirms that that particular mRNA is translating. So these all experiments could be performed to obtain transcriptome and proteomic data set. Now the next analysis what can be done is we can use statistical methods, machine learning and network analysis to compare that what kind of data we have obtained. So we can look for the functional signatures, we can check that which genes are present. So different genes have some signature sequences, we can say, which differ among different genes, through which we can recognize those genes. So we can identify those features by combining these experimental data sets. We can also look for the epigenetic modifiers, such as if there is methylation, acetylation or protein DNA interactions. So all those things can be analyzed with the help of functional signatures in analysis method, you can see. And then interaction networks, so you can also look for the genes which are interacting with one another or proteins which are interacting. So network construction can also be done. Or we can also look into the biological networks, for example the functional modules. So in our body there are certain pathways which require certain genes. So those genes will also be linked to one another. So if we disturb one gene, then that entire pathway will get disturbed. So in that case we can also look into the functional modules. So modules are the set of genes which comes together and perform a single function. So those things can also be analyzed. So these are some kind of methods which you can incorporate to your experimental data depending on what you want to analyze and what are the applications. So broad applications are, you can look into the cellular mechanisms, that in cells what kind of mechanisms is undergoing. You can also classify disease, you can identify the biomarkers, you can also look into host pathogen interactions. So these are some kind of applications, and you don't have to go into detail of these. This is just an overview, but the key takeaway messages from this paper were that these integrative analysis of transcripts and proteins, they are crucial for understanding the gene regulation, genome annotation and disease processes. Because till now we have seen that why we require proteogenomics. We have looked that we are observing the cancer data sets, that we require the information which is available in the cancer data set to design new therapeutics. So in that case, that was the first thing for which this proteogenomic was introduced, and later on it was realized that its applications are huge. So in that way, this integrative analysis will also help us to understand the gene regulations and these phenomena. And they are also saying that advances in molecular profiling and computational resources, it will also enable multivariate analysis for system level insights from this multidimensional data set. And this will also help us to combine epigenome, genome, transcriptome, translatome, proteome and metabolome to generate some kind of comprehensive understanding of biological systems. So I hope you are, so we'll discuss about epigenome because this is related to week 2. Genome is already discussed, transcriptome also you already know that the RNA which is present in the cell, but the translatome is quite confusing because the proteome is the protein which is present, but if you look at translatome, it appears that it might be a translational product, because if you look at transcriptome, so it is composed of two words, transcript and ome. So that means when you're looking at all the transcripts, means all the mRNAs or all kind of RNAs. But when we talk about the translatome, it distinguishes from a proteome in only one aspect. Translatome is the actively synthesizing protein. So when we are studying a cell and we are looking for the actively translating mRNAs in which ribosome is attached and it is actively forming the protein, then that portion of that proteins present in the cell are known as translatome. They are not referred to as proteome, because proteome is the already synthesized proteins which are present, and translatome will be the actively proteins which are synthesizing. So that will be the translatome. And metabolome is all the metabolic constituents that are present in the cell. They are referred to as metabolome, such as enzymes or other molecules, other proteins which are present. So these all are essential for comprehensive biological understanding, and this will also help us to design the precision medicine, because for example, if we study all the genome of different people, then we can identify the biomarkers or specific sequences which are specific to one person. So we can design the drugs on the basis of those sequences. So it will help us to design precision medicine as well as personalized medicine. So in that way, these are few of the applications of proteogenomics. Now I will move to epigenome. So this is the first topic of week 2. So when we say epigenome is also made up of two words. Epi means above and genome means genome. So there are certain processes which governs the expression of a genome. Those are referred to as epigenome. So epigenome consists of chemical compounds which are also known as the epigenetic marks, which could modify or mark the genome, and that will decide that what to do, where to do, and when to do. So epigenome will decide that when the gene will be expressed, when it will be silenced, and where it will be expressed. Because if you see in the human body, there are multiple, each cell have a same DNA, but the cells, so the organs, they are different, and each organ have different functionality. So DNA is same, but everything is different. So this is governed by the epigenome. It will decide that what gene to express and what not to express, because DNA is huge. There are multiple genes that are present, and not all genes are expressed at the same time. Certain genes have to be silenced at one particular time. So to govern that, epigenome is required. Epigenetic markers are required to decide that to which gene to express. So different cells have different epigenetic marks, and these marks, which are not the part of DNA itself, so they are passed on from cell to cell as the cell divides, and it also passes from one generation to next generation. So to give the introduction of epigenome, so it is the collection of all of the epigenetic marks on the DNA in a single cell. So epigenome among different cells can vary. So here in the right side you can see the structure of a chromosome. So if you all know that chromosome is a structure which is formed by the nucleosome. So what is nucleosome is when DNA is wrapped around histone proteins. As you all know that DNA is very large, it has more than 3 billion base pairs. So to accommodate that bigger DNA into the smaller nucleus, that DNA has to be wrapped around some proteins, and those proteins are referred to as histone proteins. So when DNA is wrapped around the histone protein, the structure it forms is called nucleosome, and when those nucleosomes are further cross-linked and packed inside a compact structure, that is referred to as a chromosome. So if I expand a chromosome, it will be looking like this. This is the nucleosome where the histone protein is wrapped around the, so where DNA is wrapped around this histone protein, and if you see carefully, then there are methyl groups attached to these histone proteins. So there are some genes which are also referred to as histone modifiers, which actually which modifies this histone protein. They can either attach methyl groups or they can acetylate it. So depending on what kind of modification is being done, so it will impact the expression of that particular DNA which is wrapped around the histone protein. And if you see here, it is a chromatin modeling complexes, so chromatin remodeling complexes. So I will also introduce two of the terms which might help you. So you might have heard about euchromatin and heterochromatin. So when chromosomes appear as a single thread, it is known as chromatin. So chromosome with protein is referred to as chromatin. So euchromatin is a chromatin where the actively expressing genes are present. So it is a structure where the DNA region is free. It is not compactly packed around the histone proteins, because in the chromosome the DNA is compactly packed around the histone protein. So that is, when it is compactly packed, DNA is not accessible to the other transcriptional factors and translational factors, and that is referred to as the heterochromatin. That means the genes are not able to express, whereas if the DNA is freely available, it is not compactly packed, it is accessible for replication and transcription, then it is referred to as the euchromatin. That means actively expressing DNA. So in this case also, if you see, when it is compactly packed here, then it is referred to as heterochromatin, and when it is freely available for transcription and replication, then it is known as euchromatin structure, which are also said to be the actively expressing area. Okay, so this is also the addition to the previous slide, in which the epigenetic studies the DNA changes that do not affect underlying sequence but alter the gene expression. So when previously we were discussing about mutation, so I mentioned that mutation is the change in the DNA sequence. It is not any modification, but a sequence gets changed. But in this case, in epigenetics, it means the DNA is changing, but sequence is not changing. Sequence is modified. Either some group is attached to it. For example, DNA could get methylated. So there are cytosine bases which are present in the DNA. So they could get methylated. So that is one kind of modification of nucleotide base, which is referred to as the epigenetic change or epigenetic modification. So these chemical modifications to DNA, they can decide that whether gene will be turned on or it will be turned off. And there are some epigenetic changes which can be inherited and which will also affect the gene regulation in daughter cells. So there is a concept of imprinting, So genomic imprinting, in what happens in genomic imprinting, there are certain genes which are expressed in the parental manner. For example, if a mother is giving rise to so if mother is of course we know, so if there are certain genes which are only expressed so there are certain genes which are only expressed in the maternal copy. So when a baby is formed, then it contains the genes from both the parents, but only maternal copy get expressed. So that is the imprinting, when the one of the parental gene is only expressed and other gets silenced. So that is known as imprinting. So that imprinting is also governed by these epigenetic modifications. So if you see in this figure, these are the epigenetic mechanisms and they are influenced by the development. So when a person develops, so during its early stages, there are certain genes which are expressed, but when he grows, then the proteins which are synthesized by those genes, they get changed, their rate gets changed. So development has also a role to play in what kind of expression will happen. So it will also decide. Then environmental, chemicals, so if you are exposed to certain chemicals, then it will also influence the genes and the proteins which are synthesized from those genes. Then drugs and pharmaceuticals, so there are certain drugs which will also affect your gene expression. Then aging will also affect, because if you see that the younger people or the children, they have the better immunity as compared to older people. So this is because of aging, because as the cells age, they loses their capability of replication, cell repair. So this is also one of the reason. Then diet will also influence the gene expression. And if we look at the health endpoints, that what are the health reasons that could influence the gene expression, so they are cancer, autoimmune diseases, mental disorders, diabetes. So these are few of the reasons which could influence the gene expression. And in DNA methylation, as I've talked earlier, so there are cytosine that are present in our DNA, it could get methylated. And if you know that C and G, cytosine and guanine, they are rich in the promoter regions. Promoter regions are present before the gene and they govern the expression of the gene. So if transcription factors bind to these promoter regions, so expression will be better. But if methyl group attaches to those cytosines, then the promoter regions will not be able to bind to the transcription factors, and hence it will influence the expression of that gene. So in that way, DNA methylation can govern that whether the gene will be expressed or not. So histone modification will also govern, because if histones will be modified, then it will not be able to loosen up, and hence it will remain as a heterochromatin structure. It will not allow those DNAs which are wrapped around to express properly. So these are few of the epigenetic markers. Okay, so I was telling that GC rich region is found most probably in the promoter site. So whenever genes are present, so GC rich region is present before the gene, and those promoter regions are important for binding of transcription factors. So transcription factors bind to those promoter regions, and ultimately they will they will allow the gene to express in terms of RNA. So transcription will be favored with the help of binding of transcription factor to these promoter regions, and these promoter regions are rich in CPG islands. So we also call them as CPG islands. That means C and G are rich in those promoter regions. So if there will be modification of these cytosines present at the promoter region, then they will not allow the binding of those transcription factors to that CPG region or the promoter region, which will influence the transcription. So now gene won't be able to transcribe to mRNA, and ultimately protein will not be formed. So that I was telling, that CG regions are rich near the promoter sites. So I hope it is clear now.
Yes, I've understood.
Yeah, okay. Yeah, so now I have given few techniques to study epigenetics. So this is just a brief table and I'll go to them one by one. So first technique is chromatin immunoprecipitation. So chromatin immunoprecipitation, these three terms are together and then followed by sequencing. So chromatin means if you have a whole chromosome, immuno means some antibodies might be involved, precipitation means whenever protein get precipitated, so that is precipitation. So if we combine all of them, so these are ultimately used to identify protein and DNA interactions and modifications across the genome. For example, if you want to look for the reasons in the genome where the histone is present, or you want to identify those regions where histones are more prevalent in the genome. So those regions will be inactive regions, or you can say heterochromatic regions, so that you can also identify. Along with this, you can also look for those regions where transcription factors are binding, or or other proteins are binding in the DNA. So those regions can be identified with the help of ChIP-Seq. And what is ultimately done in this process is that you cross-link the proteins to DNA. For example, if you're not looking at the histone, because histone is already cross-linked to DNA in the genome, but if you are looking for the transcription factors or some other proteins that are binding to the DNA, so in that case you have to cross-link your DNA with the proteins. Then you will shear the chromatin. Shear means you will just fragment it down. You will immunoprecipitate it. So in immunoprecipitation, you will take the antibodies that are specific to those proteins, so that it could bind to those proteins, and then you can simply sequence that protein associated DNA, because ultimately those protein associated DNA fragments will be obtained. You will remove those proteins and you will sequence that part. So at last, when you will assemble it, you will match it with the assembly or with the reference sequence. You will be able to identify that which are the sequences where the proteins are binding. So this analysis is mostly done to identify the transcription factor binding sites or some other protein binding sites in the DNA, to identify those sites. And other than this, you can study histone modifications, transcription factor binding sites, and chromatin related processes. So next technique is DNase Seq. So it is clear from the name, DNase. So that means there are certain enzymes which degrade the DNA sequence that are used. DNase, so ACE, whenever ACE is used, it means it is for the enzyme. And DNase, that means enzymes which catalyze the DNA. So DNase Seq means in this we will be using the DNase enzymes which will degrade the freely available DNA sequences, and ultimately we will sequence those regions. So in this case, you identify the open chromatin regions and identify the regions of active gene regulation. So active gene regulation regions are the euchromatin regions, in which histone proteins are not tightly bound around the DNA. So in that case, DNA is freely available, and if DNA will be freely available, then DNase can attack those regions. But if it is wrapped around a protein, then DNase won't be able to degrade. So that is the main principle behind this method. You will treat your cell with the DNase 1, and it will digest the accessible DNA where the histone is not wrapped around the DNA, where DNA is free. And then you will sequence the remaining DNA fragments, and it is used to identify the regulatory elements, study gene expression regulation, and identify DNA binding protein sites. Then comes high C, and 5C is also the modification of high C, and it is used to study the 3D organization of the genome and chromatin interactions. So we will see it in detail in the later slides. In this case, firstly you will cross-link the chromatin, you will capture the interactions between these regions, and ultimately we will sequence to map these spatial genome organizations. And this is used to understand the chromatin folding, regulatory interactions, and impact of structural variations. Then comes DNA methylation analysis, in which you identify where the methylation is happening. So investigate DNA methylation patterns, and there are multiple techniques to study this DNA methylation, but few of them I have mentioned. First one is bisulfite sequencing. So what is done in this kind of sequencing is that as we know that in the DNA only the cytosines are the nitrogenous bases which get methylated. So in this case, what we will do, that the cytosines which are unmethylated, we will convert them to the uracil by using the enzymes. So in this case, we will use the bisulfite treatment. So by bisulfite treatment, the cytosines which are unmethylated, they will get converted to uracil, and the cytosines which are methylated, they will remain as cytosines. So in the end, when we will sequence, so all those cytosines which will be in the sequence, it means they all are methylated. So in that way, we will be able to understand the methylation pattern, and it will also help us to study the gene silencing, imprinting, and cancer associated methylation changes. So if we take the sample of a cancer patient and compare it with the normal patient, so then also we can compare how the DNA methylation pattern is changing. And there is also another technique which was not mentioned in your week 2, that is ATAC-Seq. So ATAC-Seq is also used to assess the chromatin accessibility, and the difference between the ATAC-Seq and DNase Seq is only single, that in ATAC-Seq you use the transposases enzymes to insert the sequencing adapters, and in this the free accessible chromatin regions are generally digested with the help of the transposase enzymes. So that is the only difference between DNase and ATAC-Seq, and it is used to understand the chromatin dynamics, enhancer activity, and regulatory element mapping. Similarly, if we talk about single cell epigenomics, it means if you are focusing on a single cell, and for single cell you are studying that how the epigenomic markers are changing, then it is referred to as single cell. It is not some technique, but it is just mentioned because if you are focusing only on the single cell, because other techniques they focus on the entire genome, or it is irrespective of any cell. But if we specifically talk about single cell, then single cell epigenomics can be used, and for this you can either use DNA methylation sequencing or single cell ATAC-Seq. Now let's begin with the ChIP-Seq, which is the first method. So ChIP-Seq is the chromatin immunoprecipitation followed by sequencing. So as in the right figure, you can see that firstly there will be cross-linking. This is the first step where DNA is cross-linked with the protein. So in this case the example is given of the genome which is already cross-linked with the histone proteins. We are not specifically giving any protein and cross-linking it. So the second step is chromatin fragmentation. So we will fragment that chromatin because we cannot sequence the entire chromatin in a single go. So we will fragment it. Then we will do immunoprecipitation. So in immunoprecipitation what will happen is we will take antibodies which are specific to those histone proteins. They will bind these histone proteins. So if you see that it will only fragment the regions which are cross-linked to protein. So the regions of the DNA which are not cross-linked to protein, those regions are removed. We will only focus on those regions which are cross-linked with the protein and ultimately do immunoprecipitation. After immunoprecipitation, we will purify the DNA. We will remove these proteins and antibodies, and we will only obtain the DNA which is wrapped around these proteins, and we will perform its analysis. We will sequence it, and we will identify, after comparing it with the reference, that to which part of the genome it belongs. In that way we will be able to understand that what are the regions which are binded with the proteins. Okay, so this everything is written on the left side. You can go through it. And this is the workflow. If you want to look into the workflow of ChIP-Seq one by one. So first step is cross-linking and cell lysis. Second step is chromatin fragmentation, immunoprecipitation, reverse cross-linking and DNA purification. And for the library preparation, you can undergo PCR and you can attach the adapters. So it is better detailed procedure. Then you can do sequencing. For sequencing, you can use multiple methods. In this, they mentioned that Illumina is mostly favored method. And then for the DNA analysis, you can align those sequenced reads to the reference and decide that to which part of the genome it belongs. So this is all about ChIP-Seq. And there is another paper where they have mentioned it very clearly, so I have attached it. So in that they have mentioned that how the sample is prepared. So in this case they have given the example of both the modifications. You can also study the histone DNA binding sites as well as transcription factor and DNA binding site. Both those things you can study. So let's look at this table. So in the case where you want to identify the regions or the locations in the DNA which bind to transcription factors, so for that you have to cross-link with the help of formaldehyde. You have to provide those transcription factors and you have to perform cross-linking in the presence of formaldehyde. But if you want to map the nucleosome positions or the positions where histone is bound around the DNA, then in that case you do not have to do cross-linking because we know that histone is already attached to the DNA. Then comes fragmentation. So in the case of transcription factors, fragmentation is done with sonication because you are artificially doing it, so sonication can be done. But in case of the live cell where you have the histone proteins bind around the DNA, so in that case you have to go for MNase treatment. So this is a enzyme which degrades the other regions which are freely accessible, and it will only left the region with the histone protein which so DNA which is bound around that histone protein, that will not be cleaved. Rest other region will be cleaved with the MNase. Then immunoprecipitation. So in this case the antibody will be specific to that particular transcription factor, whereas in case of histone identification, so in that case the antibody will be specific to the histone modification or that particular histone protein for which you are looking for the DNA binding region. So the first process is similar. You will firstly cross-link your DNA either with transcription factor, and in case of histone it is already cross-linked. Then you will fragment it into smaller fragments. You will sequence them. then single reads will be obtained. You will map those reads. Now if we look at the second part, which is of computation analysis, so now you have mapped those reads to your reference sequence. Then you have to do normalization. So in normalization, you have to look for the confidence that this, you should be confident that this read which is mapping to one particular location in the reference, you should be confident enough to means mention that this read belongs to this particular sequence part. So these normalizations have to be done. You need as much as reads mapped to that particular region as possible to be confident about your predictions. Then peak calling is done. In peak calling, you look for the in the reference, you look for the region which is mostly for which you obtain most of the reads. So this will tell you that which of the region of the genome is mostly binding to the protein. So that will be the protein binding region. And along with this, what information it will provide you about the chromatin state annotations. So in the next slide, I will tell you that how it will tell you about the chromatin state. So as I have told you, in this immunoprecipitation step, you will take the antibodies which are specific to histone proteins. So chromatin state annotation means there are some methylations which are so in case of histone proteins, the methylation happens in the lysine amino acid. So lysine amino acid can have the methylation, and in DNA, cytosine can have the methylation. So in histone protein, there are multiple lysines present at multiple positions, and depending on which lysine get methylated, it will decide the what will be the fate of that gene or fate of that particular chromatin. So in that case, you will have the antibody specific to those histone, so those specifically methylated histones. So in that case, you will be able to identify the chromatin state, either it is expressing or not expressing. Then you can also undergo chromatin state clustering. You can look for the motif analysis, that either there are certain nucleotides which are mostly present, for example CpG island, if we call the CpG island, so it means that there is a region which is rich in C and G. So that is also a kind of motif. Motif are the secondary structures or the sequences which are seen in most repeats. So those are known as motifs. So you can analyze the motifs if you will look for these reads, so you can predict, because as I mentioned that promoter region are rich in CpG islands, means C and G are higher, and they mostly bind to transcription factors. So transcription factors are the proteins which bind to DNA. So that means promoter are the region of where DNA binds. So in that case, motif analysis can be done, and with the help of motif analysis only, it was predicted that CpG islands are mostly present at the promoter site. Then you can go for the GO analysis. So GO analysis means gene ontology analysis. In this, you look for different cellular compartment, biological processes and molecular function of those genes, that in which functions they are involved. So this gene ontology, there are already databases which are present. So whenever any experiment is performed, so that is stored in some database which is known as gene ontology database. So from there you can analyze those genes, that what kind of functionality it is involved in, where it is found in the cell. So those things can be done after you obtain the ChIP-Seq data after analysis. So now let's look at these chromatin states, that how you can predict. So there are 5 core histone marks with which you can identify that what will be the chromatin state. So this has been proposed by the Roadmap Epigenomics Consortium. So firstly is the H3 lysine for monomethylation. So it means, if we talk about histones, so there are H1 to H4 histones, there is a octet which is present, and H3 lysine 4, that means in histone third protein, there is at 4th position there will be a lysine which gets monomethylated, that means single methyl group is attached to the 4th lysine of 3rd histone. So if that modification is present, or if 3rd histone lysine 27 is acetylated, then it is associated with the enhancer regions. So enhancer regions are the one which will enhance or increase the transcription of a gene. So if this methylation happens, then it will enhance the transcription or expression of the gene. So for these two we will design specific antibodies, and if we will be able to identify the DNA sites which are binding with these histone proteins and this methylation is present, then we can predict that this particular region of DNA is the enhancer region. Then in the second histone 3 lysine 4 trimethylation. If this fourth lysine, which was previously monomethylated, if it is trimethylated, then it is associated with the promoter regions. So if this particular histone is trimethylated, then it is associated with the promoter regions, and promoter regions are the regions which are present just before the gene, and to these regions only the transcription factors bind, and then it will start the transcription and translation further on. So firstly it will start the transcription if we talk about the nucleus, and translation will begin in the cytoplasm. That will be a later process. So this kind of methylation will help you to know that this particular region of DNA is a promoter region. And if this histone 3 lysine 36, so in all there is only third histone in which the lysine are modified with the methyl group. So if 36th position lysine is trimethylated, then it is associated with the transcribed region in gene bodies. So it will tell you that this is the region which is mostly transcribed. So in this we are talking about the methylation of histones, not about the DNA methylation. So don't get confused. Then histone 3 lysine 27 trimethylation, that is associated with the polycomb repression. So polycomb is also group of or family of proteins which kind of repress certain genes. So if that particular methylation is found, then it means there is kind of polycomb repression. If third histone lysine number 9 is trimethylated, it is associated with the heterochromatin. So this is also trimethylation, and if this is the 9th lysine of the third histone, so it is associated with the heterochromatin. That means it will be a region where the DNA is tightly wrapped around the histone and that gene won't be able to express freely. So in the note I have provided that for each of these histones specific antibodies can be designed, and you will be precisely able to capture these regions present in the genome. So in this way you can identify the chromatin state, that what state it is in. Then there are some databases where you can obtain this information, and this is also present in the same paper. I have given link to that paper if you want to read. So first data set is the ENCODE portal, and it is the encyclopedia of DNA elements, and it will not only provide you the information about the ChIP-Seq, but other information will also be present, such as transcription, histone modifications, chromatin accessibility, and many more. And in the URL section I have provided you with the URL if you want to visit these portals and how the data is arranged. You can go through them. Then comes roadmap epigenome database. So this is also another database which will provide you variable human cell type and tissue data set, and it will tell you the role of epigenetic modifications in health and disease. So they are kind of integrating these data sets, and they are specific to human cell types. Then IHEC data portal. So this is the International Human Epigenome Consortium data portal. It will also provide you the high quality reference epigenomes from multiple international projects, and similarly it will also provide you with the epigenetic data set. Then comes epigenome database for human endothelial cells. So this data is specific for the human endothelial cells. It is only focusing on those cells. If you want to see what kind of epigenetic modifications are observed in these cells, so you can go through this database. So these are few of the databases, and now I have provided you with the applications. So it has already been previously discussed, so you can go through them. First is gene regulation, then histone modification mapping. There are some applications which appears to be redundant, but in the exam if you have to write the points, then it will help you. Transcription factor binding, chromatin state annotation, disease research, functional genomics, developmental biology, drug discovery. So these all are the applications of ChIP-Seq. It depends that how you interpret the results and how you integrate different analysis. So in that way you can use the ChIP-Seq in different applications in different fields. The second technique is DNase Seq assays. So DNase Seq assay in which you use the DNase. Now this assay is the DNase 1 hypersensitive site sequencing. So when we say hypersensitive sites, these are the sites present in the genome where the histone proteins, or we can say where the nucleosomes are not that tightly arranged. So but there are few. So we cannot say that there is no histone protein present. There will be histone proteins, but they are very few, so in such a way that that gene is able to express itself. So that particular DNA is accessible with the presence of fewer number of nucleosomes or the fewer proteins that are binding to DNA. So those sites are referred to as hypersensitive sites, and these DNase Seq is mostly used to identify the locations of these regulatory regions. So regulatory regions which are regulating the expression of gene based on genome-wide sequencing of regions which are sensitive to cleavage by DNase 1. So DNase 1 will be able to cleave those sites where fewer nucleosomes are present or fewer proteins are present and that DNA is accessible. So those sites can be degraded with the DNase 1, which is an enzyme. It will cleave the hypersensitive site. So now here you can see there are blunt ends of a DNA which are produced. These blunt ends will be ligated with the biotin. So biotinylated linkers. So those linkers will be biotinylated. They are attached. Then what you will do is you will use MME1, which is also another enzyme which will digest them and which will convert this longer you can say longer double-stranded DNA regions into the two base fragment. Then it will also bind to DNA dinyl beads. So there are some beads which you see here, darker browns. So they attach to this biotinylated adapters, and these beads are mostly used to isolate these two base pair fragments in the later section. And then you will go for the PCR. So in this case they are also attaching the another adapter to the end of these biotinylated fragments which are obtained. So here the blue colored adapters, they are attached to the another end. So in this case you will see the paired end adapters. So in both the ends adapters are present. PCR is used for the amplification. This biotin is removed because you will provide the streptavidin and this dinyl beads. When you will perform the amplification, they will be removed. So biotin will also be removed, and this fragment will be after PCR multiple copies will be produced, and ultimately you can go for the sequencing. And in addition to DNase Seq, there is another method which is the FAIRE-seq. So which is the successor of DNase Seq, and it is mostly used for the genome-wide identification of accessible DNA regions. So if we talk about DNase Seq, it is focusing on the hypersensitive sites. So hypersensitive sites means where fewer number of these histone proteins or the DNA binding proteins are present. But in FAIRE-seq you will look for all the accessible DNAs in the entire genome. So in that case FAIRE-seq is a modification. It is not restricted to only the hypersensitive sites. So that's why it came up after the DNase Seq, and both these protocols are used to identify the open chromatin regions, but they have biases depending on the nucleosome structure, because in the case of DNase Seq it is focusing on those regions where fewer nucleosomes are present, whereas FAIRE-seq will completely look for the freely accessible DNA sequences. And there is another paper in which this technique was clearly mentioned, so I have attached that paper. So in this they have mentioned that how the nucleus is firstly extracted from the cell, then the chromatin is obtained, then DNase 1 digestion is performed. This DNase 1 will digest the regions which are freely available, which are not tightly bound around the histone proteins. When those freely available DNA will be fragmented, then those fragments we will take it, we will attach the adapters. So we can so in the previous as you have seen that biotinylation was done, so all those things will be done. Adapters will be bound to both the ends of the DNA fragments. PCR amplification will be done, and after PCR amplification we will go for the high throughput sequencing, and we will obtain the sequence of those freely accessible chromatin regions. On the right side they have provided you how to analyze those reads. Means after sequencing, if you obtain the reads, then what can you do with it? So you can go for data pre-processing, because there are certain reads which could not be that much confident, for which you will not be confident, not that proper signal of sequencing will be obtained. So you will remove those reads. You will go for the quality control. You will map those reads to the reference to identify the sequence from where that read is coming. Data visualization will be done. So in data visualization, what you do is you take the entire genome sequence, and then the reads which you have obtained, for those you will have the peaks in the graph. So in that way you will visualize your data, that which are the regions which are freely accessible in the genome. And ultimately DNA footprinting will be done, where you will identify the significant cleavage depletion patterns in the peak regions, peak calling, peak annotations. These are some of the detailed procedures. You don't have to go through it. But what ultimately you can do is you can identify the motifs, you can go for Go annotations, and gene expression data can be studied. So in this they have looked into the transcription factor binding predictions. So transcription factors will be binding to those freely accessible DNA regions which have very fewer transcription factors which are bound to them. You can look for the transcription factor regulatory networks. You can go for the Go function analysis and integrative analysis where you are integrating all these methods together. So this is all about the DNase Seq assay pipeline, and in this I have provided you with the workflow, one by one, stepwise procedure. This will all be the same as I have discussed previously. And in FAIRE-seq, as I mentioned, that it is the successor of DNase Seq. And why it came up? Because in the DNase Seq it was limited to the hypersensitive regions. That was one of the reason. Nucleosome structure biases, so that is the nucleosome structure bias. Then comes technical limitations. So FAIRE-seq is quite simple as compared to DNase Seq, because in DNase Seq you have to be very precise. You have to identify very precisely the hypersensitive sites, but in FAIRE you are not bothered about those sites, as you are completely looking on the entire genome and checking for the freely accessible DNA. So that kind of limitation is not in FAIRE-seq. It is higher resolution because not just the promoter site, but you will also be able to look into the known promoter regulatory elements. And then cost and resource. So the cost of FAIRE-seq is low because, as I mentioned earlier, that you don't have to be specific for the hypersensitive regions. So these are few of the points that why FAIRE-seq came up and what are the additional plus points of FAIRE-seq as compared to DNase Seq. So till now everything is clear, or do you have any doubt in some section? Are you there? Okay, thank you. Because there are very few people in this session, so I thought maybe you are also not there. So okay, so now in this slide I have given the comparative analysis of different methods, that when to use which technique. So in case of, if you want to study the so when you use ChIP-Seq, so when you want to study the chromatin and protein interactions, so in that case you use ChIP-Seq. But in all these methods the common procedure is that you cross-link the DNA binding proteins, you go for the fragmentation of DNA and immunoprecipitation, DNA is purified and sequencing libraries are prepared and data is analyzed. So this is kind of general overview of all these methods. But in ChIP-Seq you look for the open chromatin, you go for sonication. So open chromatin here means that you open the chromatin first and then you check for the histone proteins which are attached to the DNA regions, and then you go for the fragmentation, sonication, immunoprecipitation and DNA purification. Similarly in case of DNase Seq you have open chromatin. Instead of fragmenting it, you go for the DNase digestion, and then the freely accessible DNase will be available. You will fragment those fragments will already be obtained. You will go for electrophoresis where you will separate those bands on the gel, and then you will purify that DNA and sequence the library. In case of ATAC-Seq, as I mentioned, that in case of DNase Seq you use DNase, whereas in case of ATAC-Seq you will use the TN5 transposase. So transposase are the enzymes which are mostly used in the transposition, and transposition is a phenomena where in the gene there are some internal segments. So in the genome there are some segments which can act as a jumping gene, which can change their position. They can cut from this position and they can anneal to another position. So that is known as transposition, which is like a cut and paste mechanism or copy and paste mechanism. So in this case, if we talk about cut and paste, so cut means that some fragment is cut from one side, it is pasted to other side. So that is transposition, and that transposition is held or means done with the help of transposases. Okay. Yeah, so that is facilitated by these transposases. So in this case, the transposases will come, they will identify the freely available DNA sequences and they can cut it. So this is the main difference between DNase Seq and ATAC-Seq, and rest procedure will be same. You will attach the adapters, PCR will be done, and at last sequencing will be done. Then comes MNase Seq. So this is also like DNase Seq only, but in this the enzyme which you are using is micrococcale nuclease. So it will also degrade the freely available DNA sequences, but it is obtained from the micrococcal organism. So due to which its name is DNase Seq, and similar procedure will be done. FAIRE-seq is also like DNase Seq, and it is for the entire freely accessible genomic regions for the entire genome. So this is a comparative analysis. Now let's move to the next technique, which is the high-C, and high-C is referred to as 3C, which is the high resolution chromosome conformation capture. So high-C can be of multiple types, multiple variants could be there, but mostly high-C is the 3C method. And now the later on advanced stage is 5C. So we will see that how these C varies. So this 3C is coming from chromosome conformation capture. This high-C method is used to capture the conformation of chromosome, that how it is arranged, where are the histone proteins bound to the DNA. So those things, those conformations can be captured, and it maps the spatial organization of the entire genome, revealing the interaction between different regions of DNA. So what is the principle behind this technique is that if some DNA is wrapped around histone protein, so what histone protein can do, it can bring the genes which are present far to one another closer, because when that DNA will wrap around the histone protein, then the gene which is lying very far away, that can also be bring closer to the gene which is present on the upper end of the histone. So in that case, two genes will be brought closer with the help of the histone protein. So in that case, those genes which are residing close, it is believed that those genes will share some kind of functionality, or they could either help one another in the expression. So that's why the high-C came up, that we can identify those genes which are lying closer to one another to check that either they have some kind of interaction with one another. So means that can also be done with this high-C. So it will help you to identify the spatial organization of your genome, that how the genes are organized. And in this technique, you will cross-link the DNA with the formaldehyde, cut it with the restriction enzymes. So in the right side, if you see this figure, so they have looked into the chromatin interactions in the nucleus. These are multiple cells. They are focusing on a single nucleus. If you see, the DNA is wrapped around this histone protein. These are the histone proteins. Now they will cross-link the DNA. So in this case, they have provided some protein to cross-link the DNA, because cross-linking means it will bring the DNA closer to one another. So cross-linking will be done. Histones are separated. After cross-linking, they will digest it with the HIN3. So whenever HIN3 digestion will be done, these are the restriction enzymes which will generate the sticky ends, and whenever sticky ends will be generated, it is easier to ligate them back together. So after HIN3 digestion, they will biotinylate these ends. After biotinylization, so after biotin being attached, next step is the proximity ligation. So if the two DNA, if they are present close to one another, then they will be ligated. If they are far away from one another, they won't be able to get ligate together. So in this case, proximity ligation, they will ligate, they will form this kind of structure. And the cases where they are not able to ligate to one another, the cases where they are far away, those DNAs are far away from one another, so in those scenarios, they will not cross-link. So we will remove them with the help of T4 DNA polymerase. They will remove those unligated fragments. Now after the ligated fragments being obtained, we will extract them, and we will go for the sonication and repair, where we will, from cross-linking, we will convert them into the straight DNA single-stranded fragment. Biotin will be pulled up by the help of streptavidin and other proteins. They will remove the biotin from these fragments, and ultimately we will go for the paired end sequencing, in which we will attach the adapters, or we can use the probes, which will, or the primers, which are you can say which are the independent primers which can be used for the PCR amplification of those repeats or those sequences which we have obtained, and ultimately we will be able to find that which of the genes are lying close to one another, because they will only get cross-linked. Otherwise, if they are far away, they won't be able to cross-link. So it will offer us the unique insight to transcription regulation and cancer development. So we will see in the later slide that how it is helping. So applications, if you see here, Hi-C provides insight into the chromosomal architecture, long-range genomic interactions, and formation of chromatin loops and domains. And it also helps in understanding the role of chromatin organization in gene regulation, and can also reveal the structural changes which are associated with the disease. For example, if you are comparing it, then you can draw insights after comparing the normal state and the cancer state, that how the chromosome is organized, and in that case you can relate that if chromosome is arranged in this form, then it is leading to disease. So for this, high C is used. Now in this case I have talked about series of high C. So beginning with the 3C, which is chromosome conformation capture. So it measures the interactions between a specific pair of genomic regions by cross-linking, digestion, religating chromatin. Religating means with the biotin being present and they are religating together, proximity annealing, and then quantifying the resulting chimeric DNA. So chimeric DNA, why? Because I have told you that the histone is kind of bringing them two far away genes together. So if two genes are combining, two different genes are combining, it is known as chimeric product or chimeric DNA. So you will be able to quantify the chimeric DNA. If we talk about 4C, it is the chromosome conformation capture on chip. So if you target the interactions of specific genomic region with the rest of the genome, when it provides you the details of the interactions related to particular genes or regulatory elements, then it is referred to as 4C. When you are only focusing on the certain genes and you are looking for those genes, that which other genes are related to it. For example, P53 is one of the gene which is mostly observed in different types of cancer, and now I want to see that what are the other genes which are linked to it, because P53 is also observed in the normal tissues, and I want to find some target or find some gene for which I can design a drug. It could silence that gene and it could control the disease. So in that scenarios, I am only focusing on the P53 gene. So in that case, I will use 4C to check which are the other genes which are closer to it. So that for that purpose you can use 4C. Then comes 5C, which is the chromosome conformation capture, carbon copy. So it is used when you are going for the multiplex probes to measure the interactions between many of the genomic regions, simultaneously offering detailed map of chromatin interactions across larger regions. So when I am focusing on the entire genomic region and I am having multiple samples, so in that case I will go for the 5C. And then there are some other of series. If you want, then you can go through them, such as high C with the long reads, in situ capture, single cell resolution, and cross-link covariance. So these are not in your syllabus, but if you are interested, you can go through these slides. And I think 3C, 4C, 5C are enough. Okay, then there was another paper in which it was mentioned that why 5C came up and how it is helpful than 3C. So in this paper also they have mentioned that 5C is actually allowing you to do for the simultaneous analysis of multiple chromatin interactions. So not just a single one gene interacting with other gene at the one particular section, but you can also look for the multiple such interactions at the same time. So for that you can go for the 5C. So that is one of the major advantage of 5C. Now let's come to our third or the fourth technique, which is the DNA methylation analysis, in which we will be looking for the sites in which the DNA is methylated. So DNA methylation is a heritable epigenetic mark which involves the covalent transfer of methyl group to the C5 position of cytosine ring. So this can come in your competitive exams, that where the methyl group is attached in the cytosine ring. So it is the 5th carbon of the cytosine ring where the methyl group is attached, and here it is. So, and it is very important to understand these methylation, because these methylation patterns will govern that how the DNA will be expressed, and it will also decide the role of DNA during the development and differentiation. Because if there is abnormal methylation pattern, so it is mostly observed in the cancer. So that's why it is important to identify the methylation marks. If you have the cancer patient, then you can compare his methylation marks with the normal person to predict that whether he is suffering, he is at what stage, or what could be the drugs which could be designed. Or for example, if you don't know that that person is suffering from cancer or some disease, so you can predict that if whether he will suffer, or his stage could be identified with the help of DNA methylation patterns. And I'm not saying that by just looking at the methylation patterns you will be able to comment, but it will help to strengthen your studies, because you can integrate multiple other analyses also. So this could be one of the analysis which could be done. So there are different methods to do this DNA methylation analysis. One such method is the whole genome bisulfite sequencing, in which for the whole genome you go for the bisulfite treatment. So in this, how you analyze the DNA methylation? Firstly, you will use the bisulfite. So when sodium bisulfite is used, it will convert the unmethylated cytosins to the uracils. So what it does is it removes the amino group which is present on the cytosine. So after removing the amino group, that cytosine will get converted to uracil. So amino group is removed by the sodium bisulfite, whereas if cytosine is methylated, then sodium bisulfite will not be able to attack on that methyl group. So in that scenarios, cytosine won't be converting to uracil. So now you will be able to distinguish between the methylated and unmethylated cytosines with the help of this sodium bisulfite treatment. You will prepare your library. Library preparation is done by fragmenting those bisulfite treated DNA, and then you can attach the adapters. You can go for PCR to increase the number of sequences, so that you can, means in the end if you sequence it, so you have more number of reads. For that you can go for the amplification. And then after sequencing, you can use for sequencing you can use high throughput sequencing methods. For example, Illumina is mostly cited in every place, but it depends on you, so what kind of analysis or what kind of method you want to use. So after sequencing, you will analyze. So what you will analyze from the sequence, that in all the positions where uracil is present, it means those cytosines were non-methylated. So in that way you will able to identify the non-methylated cytosine sites, and where the cytosine is present, you will be able to identify the methylated sites. So in that way you can compare the methylated data set among the normal and disease patient, or you can simply compare the normal individuals, and you can see that whether in normal population also the methylation pattern varies among people. So this can be done with in whole genome bisulfite sequencing. Then there is another method which is a reduced representation bisulfite sequencing, and this method is like cost effective, because in previous method you are completely using sodium bisulfite, which could be quite costly. So in this case it is cost effective method, in which you are focusing on the CP rich regions of the genome. You are not focusing on the entire genome, but you are specifically targeting the CPG rich regions of the genome, and you will use the restriction enzyme digestion along with the bisulfite treatment. So firstly you will use the restriction enzyme. In this case they have used MSP1. So with MSP1 the digestion has been done. Firstly DNA is isolated, then digestion is done, ends are repaired by using the adapters, and then adapter ligation is done. After adapter ligation, those small fragments we will go for the bisulfite treatment. So in bisulfite treatment, similarly as in the previous method, the cytosine will be converted to uracil, and the methylated cytosine will remain as methylated cytosine. Then those fragments will be selected, PCR could be done, sequencing could be done. So this method is same as the previous method, but the difference is that it is focusing on the CPG rich regions, and second thing is it is using the restriction enzyme before directly treating with the bisulfite. So that's why it is quite cost effective. So this is the only difference between both of them. Okay, so now I have provided here the comparative analysis just to remember all these things which we have studied so far. So in this, if you look at the right side figure, so if you want to study DNA methylation patterns, so which techniques you can use? You can use whole genome bisulfite sequencing, reduced representation bisulfite sequencing, then MEDIP, which is also the methylation DNA identification protocol sequencing, methylation arrays. These all techniques can be used to study the DNA methylation. If you want to identify the transcription factor binding, then you can go for the ChIP-Seq, ChIP-Exo, ChIP-mentation, cut and run, cut and tag. So these are some other techniques through which you can go and identify the transcription factor binding sites. If you want to look for the histone modifications, then you can use ChIP-Seq, ChIP-Exo, ChIP-mentation, similar techniques which were mentioned in case of transcription factor binding. Then chromatin accessibility, if you want to see that which regions are accessibly available or which DNA is freely available, so you can go for the ATAC-Seq, DNase Seq and FAIRE-seq, so which we have studied. Then chromatin conformation. So if you want to look at the conformation of chromatin, which genes are lying close to one another, which are far away in the three-dimensional structure, so for that you can go for the Hi-C. And then there is some another technique which is the CHIA PET. So this is also some kind of technique. So we won't go into deep of these techniques, but this is an overview that these kind of analysis can be done with these techniques. And in the left side also I have provided that if you want to study the long range regulatory elements and the cis regulatory elements, then you can use DNase Seq, FAIRE-seq and ChIP-Seq. So long range interactions and cis regulatory elements. So what does it mean is, when we talk about the long range regulatory elements, first of all, regulatory elements, that means which are regulating some process. So in this case, regulation means the transcriptional regulation, or you can say which are helping in expression. So long range means which are lying far away from some gene and then they are controlling expression of that gene. So these are also referred to as the transacting elements. These are enhancers, repressors, silencers and insulators. So these lie far away from the gene and control its expression. And to study these we use the trans-QTLs, so which we will talk in the later part of the slides. Then comes cis regulatory elements. So when we say cis regulatory elements, these are the short range interactions, so which are lying closer to the gene, such as promoter regions, which lie next to the gene coding region, so which influence the expression of the gene. So these are the cis regulatory elements. So these both the elements can be identified with the help of DNase Seq and FAIRE-seq and ChIP-Seq, because ChIP-Seq will help you to identify the protein binding sites, and these regions, they bind to the transcription factors or some activators. Activators are also the proteins. So in that way ChIP-Seq will help you to identify these regions. And then comes if you want to look for the which transcripts are being expressed, so you can go for the RNA-seq. And computational predictions and RT-PCR can be done to increase the number of copy number of the DNA reads which you have been obtained. And then in 5C, as they have shown, that for the chromosomal orientation or organization, you can go for the 5C, that how it is arranged, how the chromatin is arranged three-dimensionally, what is the conformation. So this is all about these techniques. So till now, is everything clear? I think I have went into deep detail. Okay, so after the completion, when I'll be uploading these slides, in the end I'll provide the link for every technique, the video link to every technique, don't worry. Okay, so now the next topic of this week was the TCGA, which is the Cancer Genome Atlas. And if you look, then this is the home page of Cancer Genome Atlas when you search it. So it is under the National Cancer Institute who is hosting it. Okay. So Paulami, I could not understand what are you asking. Yeah, I'll be providing you this PPT. So I hope you have accessed my previous PPT also, because to access the content, what you have to do is on the NPTEL site only. In the end, I think they are giving you link to the problem solving session. When you click on that, then you will be able to access a spreadsheet in which the link will be present to the video also, to the YouTube also, and to these slides also. There will be a drive. But along with this, what I am doing is that for every YouTube video I am attaching the link of the slides. So in case if you are not able to access it from the Swayam portal or the NPTEL portal, then you can simply subscribe my YouTube channel, or simply go to my YouTube channel and click on the video, and in the description you will be able to get the link to the slides. So if you won't be able to access it, then do let me know. And if you want to find my channel, then you can simply write my name also, then also the channel will come. Don't worry. Okay, so this is the home page of Cancer Genome Atlas, and it is under the NCI, which is the National Cancer Institute, hosted by the Center for Cancer Genomics. So as we discussed in the previous session also, that there is a GDC, which is the Gene Data Commons. So in that they have arranged the data from multiple experiments, and TCGA is also under those consortiums. So that's why if you click on the access TCGA data, which is present in the bottom part of the slide, so it will take you to it will take you to this portal, which is the GDC data portal. So don't get confused. So TCGA is also hosted by the GDC data portal. So it is in the collaboration. So they all collaborate. So if you go to any of these datasets, any of these databases, you will be redirected to the GDC data portal. So before moving to GDC data portal, I will give you a brief introduction about the Center for Cancer Genomics, who is hosting these projects. So the aim of this Center for Cancer Genomics was to take the experimental data from multiple sources, combine it, integrate it, and provide the better insight to the normal people, so that everybody could be able to access that data and perform the studies. Because cancer is of the major concern, so we need to design new drugs and new things. So in that case, the collaboration has been done so that everybody should be freely able to access the data set, so that it could help the humankind to design the better therapeutics. So in this case, its work in genomics is majorly categorized into genome sequencing, functional, and computational. So in this case, their main aim is to characterize the mutations and other molecular features of cancers. They are focusing on the cancer, not other diseases. Then in terms of function, they are looking into the biological pathways which are involved in particular disease. For example, apoptosis is also one of the pathway which is a programmed cell death. So if programmed cell death won't be properly maintained, then what it will do, the cells will continuously keep on dividing. Apoptosis won't happen, it will lead to a tumor, which is also further, if it is malignant, then it will spread in the entire body. So it is also the cancer phenotype. So these studies can be done with the help of functional analysis, and in computational, they are improving the data accessibility and analysis. And then I have also attached that what kind of questions they are answering. So there are these many questions which they are trying to answer. So I'll discuss few of them. So first one was that what genomic profiling methodologies are able to resolve cancer into subtypes that are biologically distinct and respond differentially to therapies. So the first question was that there are some cancers which have different subtypes, and each subtype is different. They behave different to different drugs. For example, if I talk about the stages of cancer, so other than subtypes, if we talk about cancer stages, then some stages are normal. When it is a first stage, then certain drugs could be used and it would be able to treat that cancer. But if it is the second stage, then we require some high efficiency drugs. In third stage, some other drugs are required. So similarly, if we talk about subtypes, so each subtype is different, and different drugs act differently on different subtypes. So their aim is to distinguish between the subtypes of cancer, and then for that particular targeted therapy could be designed. Then second question was that what new technologies for the molecular profiling of tumors can add to our understanding of cancer biology. So they are also looking into what kind of molecular profiling could be done. For example, previously we talked about the DNA methylation analysis. So now we know that methylation is differentially done in the normal state and cancer state. Similarly, if we perform more and more analysis, then we can see that what other patterns or what other molecular profiles are varying among the normal and cancer patients. So in that case, we will be able to identify the person whether he will suffer from cancer, or to predict or distinguish between two people. So in that way, the technologies could be designed. And then the third is that given the heterogeneity of cancer between the patients and the heterogeneity of cell types within individual tumors, what type and scale of molecular profiling is needed to improve the diagnosis and treatment of cancer. So for example, in cancer also, firstly there are multiple types of cancer which is present. Then there are some cancers which are solid tumors which are fixed to one place, but there are some other cancers which are blood cancer or some other cancers. So they are freely moving, they are movable. So in that cases also we need to identify that which are the tissues which are influenced and what kind of therapies should be designed. So we need to profile them separately. There are different diagnostic methods which has to be used, different treatments. So to sum up all, these all are the questions of the CGC mission, and in this they are trying to answer everything, these questions, and they have not met all these questions, but their aim is to answer these questions. So now let's move to GDC genomic data commons. So this is the data portal which you will see when you click on the access TCGA data on the previous portal. And in the previous session also I have shown you this, that there are 83 projects. These are the primary sites from where they have collected the samples and they have analyzed it. These are the cases, 44,000, 1 million files are present, 22,534 genes. For these genes they have the information in the cancer, and these are the mutations, about 2 million. And in this case they have provided you different tissues and what are the primary sites, so major primary sites. For example, in this case bone marrow and blood. So for that they have many cases, which means that blood cancer is most prominently observed as compared to other cancers, such as bile duct cancer is very less cases are observed. Okay, so now I will tell you that what you can do is, so now if you go to this portal, when you will scroll down, then you will see this, which are the core tools, in which you will see projects, cohort builders, repository. I have provided you the link, so in case if you are interested, you can explore this portal. You can click on the links and you can go through. So in this few things I will show you. So for example, if you go through the projects, then you will see these projects. So on the left side there are multiple things which you will find, such as primary site. So in primary site there will be multiple tissues which you can select. So in this case I have selected brain, but other than brain there will be multiple tissues which will be present, such as lung, breast, bile duct, stomach. There will be many tissues which will be listed. Then in case of program, these are the 3 programs which are collaborating here. So CP-TAC, HCMI and TCGA. So I have selected TCGA. Then in disease types, so depending on what primary site you select, there will be different disease types. So in this case, gliomas, mature B-cell lymphomas and not reported, these options are present. I have selected gliomas. Then in data category, you can select that what kind of data you want to analyze. For example, in this case I have selected DNA methylation. You can also look for the copy number variations, proteome profiling, etc. Then in experimental strategy also, there will be multiple experimental strategies which will be shown. Depending on your interest, you can select any one of them. So for example, I have selected DNA methylation. So in this case I am selecting methylation array. So in this way, after selecting everything, on the right side you will see that how the data will change. So in this case now only 2 projects are being shown, and disease types are the gliomas, primary site is brain, program is TCGA. And you can also, these are the check boxes, which means you can select multiple options as well. So now I have selected only these few options. So out of them, if I click on the first project, which is TCGA GBM, then I will be taken to this slide. This window will be opening. So this is the project ID, and if you see carefully in the cases and file count, data categories are biospecimen. It will tell you that what are the number of cases which they have obtained in case of biospecimen. So 617 cases are present and 2920 files are present. For copy number variations, 599. For clinical data category, 617 cases are present. For DNA methylation, 423 cases are present. So these cases are the how many methylations are being observed. So these are the cases and these are the files. So in this way, there are multiple data categories. You can go through them. Whatever category you want to explore, you can explore any one of them. Similarly, on the right side, they are telling you which of the technique being used. So in ATAC-Seq, there are 9 cases, and diagnostic slide 389. Similarly, you can go through them, such as RNA-seq, etc. If you scroll down, then you will see in the annotations, they will also annotate this. So in this case, they are giving you the information of the somatic mutations which are associated. So there are multiple cases, and you can go through any one of them. So in this, there are 3882 annotations which are present. This is a huge data. So instead of going to all the annotations, I have selected the first case. So in first case, if you click, then it will open like this, in which it will give you the information about this particular case. Similarly, biospecimen, clinical, copy number variations, DNA methylation, similar data categories will be provided, and similar experimental strategy information will be given. If you will scroll it down, then you will be able to, in the biospecimen, there will be other samples which will be provided. So actually this database is very huge. So you have to click on links, then links, and multiple links will be there. It is a data repository in which data is embedded. So in samples, in biospecimen, if you click on this sample, then ultimately it will open up the most frequent somatic mutations, from where you can see that which are the genes which are expressed in the brain cancer. So in the brain cancer, you see ZNRF3 gene, in which there is a protein change. So these are the list of the somatic mutations which are happening. So in the first mutation, you see R245 star, which means that R, which is the arginine amino acid, it is replaced with star. Star means now it is a kind of stop gain mutation, which is the missense mutation. sorry, not missense but nonsense mutation, where the mutation is such that the stop codon has come and protein is prematurely terminated. So which means that previously arginine was present, now it is leading to the formation of stop codon, so arginine is not formed, and then protein will not be translated further, it is terminated at this site. Similarly, other somatic mutations also you can see. For example, in the second you see that the threonine is replaced with the isoleucine, so it is a missense mutation where the amino acids have been changed. Are you not able to see my cursor? Oh, okay. So I don't find any option to turn on the pointer. Okay, maybe from next session I'll look for the pointer, but now I'll say in the way that you will be able to follow. Actually, I'm not able to find on the bottom left. There is the option for whiteboarding, but it will open up the whiteboard. Maybe on the Zoom it is available, but in case of Google Meets, okay, I don't know. Okay, I'll see in the next session if I'll be able to find. So okay, so in this slide, if you see the second row, if you see in the second row, the threonine is converting to isoleucine. So are you able to follow this table? So in the table, if you look at the second row, second data entry, in which the T-span 16 gene threonine 123 is converting to isoleucine, this type of substitution is a missense mutation, in which, so there is a substitution, that means some nucleotide has been changed, so this is a substitution, and it is such a substitution which is leading to missense mutation, in which threonine is replaced with the isoleucine. Okay, so similarly there are some other mutations which you can find here. So in this way, in TCGA you can firstly look for the particular tissue, and there are multiple fields which you can select, and ultimately you will be able to find somatic mutations, and there are other things also which are present. So in this way you will be able to get that which genes are involved and what kind of mutations they are undergoing. In the right side, if you see number of affected cases, which is the second last right side of the table, so in this category there is number of affected cases across the GDC. So in this they are telling you that in how many such cases this particular mutation was observed. So in the first case, if you see, it is the 6 by 16,477, so its percentage is 0.04%, which is very very less. So that means that this mutation is not frequently observed. So our aim is to look for those mutations which are frequently observed, because those frequent mutations will help us to confidently say that this particular mutation is involved, and this is a rare mutation, which means it is not majorly observed. So we cannot assure by looking at this mutation that it is involved in brain cancer or brain tumor. And similarly on the rightmost end, if you see impact, so it tells you that what is the impact of this particular mutation. So in this it is written as HI. HI means high impact, so which means that although it is only observed in 0.04% cases, but it has a high impact. That means it could lead to the death of the individual. So that could also be one of the reason that we are not able to observe it in multiple cases, because either those individuals die in the early stages, that it is such a violent mutation, so it is a high impact. Similarly in the second section, if you see, it is the MO means moderate. Similarly TO is tolerated mutation and B is benign. So these are some of the categories which are given to these mutations, that either these mutations are having higher impact, or either these are moderate, or either these are tolerated, that they are not that much influential, or benign, that means it's not influencing a lot, and some are malignant, which are leading to the tumor, which are tumor specific. Okay, so that's it about the somatic mutations. I won't go into the detail. If you want to explore, you can go through the TCGA server and see. So this is the stepwise procedure. So you can simply click on this link, which is the GDC portal link, and it will take you to the TCGA data portal, and it is combining the data from 3 different portals, so TCGA also one among them. You can select the cancer which I have shown you, then you can select the data, that what kind of data you want to get, clinical data, genomic data, transcriptomic data, depending on the need. You can also download the data about these. So in case you want to download it and analyze it separately, because in TCGA you cannot analyze it. It provides you the data, but it do not analyze it. So you can download this data and further process it, integrate it and analyze it, for which you can use statistical analysis methods. You can visualize it with the help of heat maps. So in heat maps you can take different genes, you can compare the mutation rate of certain genes, and you can also, for example, there are more missense mutations being observed or more nonsense mutations being observed. So depending on that you can compare, but this all analysis you have to do by yourself. TCGA will just provide you with the data. So to simplify this, on the further stages there comes a cBioPortal. So this portal, the main aim was this portal is to simplify the analysis. So what it does is it takes its data from TCGA and it performs its analysis. It will provide you the analytics of the multiple studies which have been already performed. So it is a user friendly interface, because in TCGA, as I have shown you, that there are links within links. You will get lost when you will be searching for the data. So for that you can simply go for the cBioPortal, which is user friendly. It integrates the data explorations. It has advanced visualization tools. In one place only you will be able to visualize multiple things, and we will see that how to use cBioPortal. It has already predefined queries and their analysis, and it provides the accessibility to the data, so you can simply download it. So now I will show you that how cBioPortal looks and how you can look for the data, because in your assignment also you have a question. So this is the portal, cBioPortal interface. So whenever you go to this cBioPortal, you will see like this. This is the home screen. Now on the left side, if you see, there are multiple studies which are shown, and on the middle there are different studies which are available, different studies which are available. So you can select any one of them, such as pan cancer, pediatric cancer studies. So you can select one among them, and there are some samples which are used in these studies, so which are also provided towards the right side. And in the bottom, if you see, there are two options. One is query by gene, and another is explore selected studies. So either if you know some gene and you want to see that in which cancers it is involved and how it is involved, and you can also do survival analysis by using cBioPortal, that if this gene is methylated or if this gene is mutated, what is the probability, what is the survival of a person. So you can simply go for query by gene, and you can also enter multiple genes also. Then comes explore selected studies. So in this case, if you are selecting some kind of study, then it will involve multiple genes. So you can also, for example, I only want to study the brain cancer or the brain related cancer. So in that case I will go for explore selected studies, because I don't know which gene is involved. Now I will give you one example. So in your assignment you were given to look for the ampulla of Vater. So in the left side also you will be able to find ampulla of Vater, which is having the one study. But for the example I will take CNS and brain study. So firstly you will select the CNS and brain study. So I hope you will be able to see these numbering. So firstly we will select the CNS brain. Then on the middle section we have to select any of the study. So for means my desire, I have selected the CNS brain cancer given by CP-TAG GDC. It depends on you which study you want to see. So it has 74 samples. So secondly I have selected it. Now I will go for explore selected studies, because I don't know the gene. So I will explore the selected studies. Now after explore selected studies, it will give the result in this form. So let's start with the left side. So this is the summary which is opened up. So in summary, firstly they will give you the genomic profile sample counts. So it tells you that what is the molecular profile. So firstly mutations, how many number of mutations are observed. So there are 74 samples in which we have seen, And in 74 samples, in all the 74 they have looked into mutations, and frequency is 100%, which means mutation is observed in all the samples. Then mRNA expression Z scores, then mRNA expression TPM Z scores. So there are multiple molecular profiles that how the transcription or expression is changed, and similarly number of patients, number of samples which are showing this change will be given, and frequency will be given, that what is the frequency of that mutation or frequency of that expression. And then let's move to the second section, which will give you the KM plot. So these are the plots for better visualization, that disease free and overall with months, and disease free. Then in the second section, if you see, there are mutation counts. So how many mutations are observed? So these are the number of mutations versus number of patients. So if I say mutation, which means in one particular cancer, it is not that ki one person will have only single mutation. They could have multiple mutations. So which is referred to as frequency of mutation. Some, for example, 2 patients are having 10 mutations. So that means the 2 patients are having, the frequency of mutation is 10. So similarly, the mutation count and its frequency is given. Diagnostic age is given, because there are some cancers which are observed in certain age. There are some cancers which are only observed after 30 years of age, or there are some cancers which are observed in the smaller infants. So in that case, the diagnostic age is given, that when it was diagnosed. And then another analysis in the middle, you see mutated genes, 74 profile samples. So in this case, they are giving you the list of genes which are observed, and then the number of mutations which are observed in these genes, and what are the frequency of these mutations, and how many patients this mutation was observed out of 74. So similarly, you will be able to look into mutated genes. Then the second to mutated gene is the CNA genes. CNA genes are the copy number alteration genes. This information will also be you will get from this cBioPortal. Then let's look at the rightmost section, which is the number of samples per patient. So how many samples were taken from one patient. Then the second to this is overall survival status. So below this is overall survival status. So in your assignment, you were asked that you have to tell number of patients which are surviving or which are dead. So to check that, you can go for the overall survival status. You can click on it, and then it will open up like the bottom one part which I have shown, which is the overall survival. So this arrow is projecting you to the how it will open up. So if you click on overall survival status, it will pop up and it will open like this, which is shown in the bottom part of the slide. So it will tell you that number 1 is the deceased and 0 is the living. So deceased means how many have died. So in this, out of 74 patients, 57 have died of this due to this in this cancer from which the sample was collected, and living are 16. So in this case, all these analyses have been already done at one place. You can simply go and analyze it. So this is the advantage of cBioPortal over TCGA. So instead of going to TCGA, you can simply go to cBioPortal. It will make your analysis easier, and there are multiple things you can explore it. So I will provide you the link. Okay, so in the latest slide, so I have also, if we scroll down and if we look at the clinical data, so previously we were focusing on the summary. If you see left topmost region, so firstly we were focusing on summary. Now if we click on clinical data, then this kind of table will open up, which will give you all the informations. And for example, if there is some cancer which have multiple subtypes, so that subtype information you can obtain it from the clinical data. So in this case, we do not have any subtypes, but in the assignment they have asked you the subtypes. So for subtypes, you can go to the clinical data, and in the one of the column you will be able to find subtype. And now just in case if you don't find the subtype in these columns, then what you can do is on the rightmost part you see the custom selection and then columns. So in columns, if you click on these columns, then you will be given the list of columns. So you can select that which column you want to see, because there are multiple columns. Some columns might not be required, so you can simply select the columns of your choice. So in that case, if subtype is not shown, so from here you can select the subtype column, but otherwise it will be shown to you. So in that you can look for the subtypes. In this case, we do not have any subtypes, but other than that, we can look for the number of samples per patient, mutations count, age of diagnosis, survival status. So all these things can be seen from this table, and we can also download this table. So if you see topmost right section where the 74 samples are written, so towards there are 3 sections in which the third option is the download option. So you can download this data also. Then if you look at the plots, so in the plots what you can do is you can give a gene of your interest. So which gene you want to observe. In this case you have to give gene by yourself. It will not have gene of its own. So to plot you have to give the gene of interest. So in this case I have given IDH1. You have to select the data type. In this I have selected copy number. You can select mutation also, and copy number profile also you have to select what kind of alteration you want to observe. So in this I am looking for the IDH1 gene, which was majorly mentioned in literature that it is involved in the brain cancer. So I am looking towards its plot. So one is diploid, one is gain, and now they are also showing that which kind of mutation is majorly observed: missense, non-mutated, or gain mutations, or diploid. So this is the plot which you can see. In this I have when I tick on mutation type and copy number, which is on the right side, then it will show me both these in the legend both these things, and in the plot also both these things will be highlighted. But if I only select the mutation type, so rightmost section of my slide, if you see, if you only select the mutation type, then you will only see the mutations which are majorly observed. For example, in this, not mutated is widely observed. Mutated missense mutation is very less. And so it is the mRNA expression to the copy number alterations. So if you see, missense is present near the 4k, below 4k only, which means that mRNA expression is less than 4k when this mutation is involved. Yeah, so these plots will be shown to you from this plots section. So ultimately you can do multiple things with the cBioPortal. I won't go into detail. Now I'll quickly talk about the SNPs and copy number alterations. So SNPs are the single nucleotide polymorphisms in which only a single position or the single nucleotide is changing, and which is changing across the DNA sequence among different individuals. That's why it is referred to as single nucleotide polymorphism, because multiple DNAs are compared and only single nucleotide is changing, rest everything is common, so it is polymorphism. And then it could occur in coding regions or non-coding regions or intergenic regions. It could be present anywhere. We are not restricting it to only coding regions. And then it is also used in the genome-wide association studies to identify the genetic variants which could be associated with the disease or traits. And it's not always that SNV will lead to a disease. It could also help in certain traits. So these all comparisons can be done in GWAS. And it also influences that how the individual respond to medications. For example, when we talk about the precision medicine or personalized medicine, then we can focus on these SNPs because different people have different SNP profile. So on the basis of that, we can design the personalized drug for them. Now let's talk about the copy number alterations. So copy number alterations, that means there is change in number of copies. So whenever gene copies are changed, the number of genes are changed, that is referred to as copy number alterations. That means for example, in the right side, if you see, the middle chromosomal part shows that there is a gene which is highlighted in the yellow color. So if this gene is lost, so in copy number alteration you can either lose the gene, which is the deletion. So right side shows the deletion event, so this gene is being deleted. Now chromosome is not having that gene. Whereas on the left side, if you see, there is the amplification. Amplification means the number of this the copy number has increased, amplified, it has doubled. So now there are 2 copies of this gene which is present. So this is referred to as copy number alterations, which can result in gain or loss of the genetic material. And these copy number alterations can have significant effect on the gene expression and function, and it could potentially lead to diseases such as cancer, because if more genes will be present, then more protein has to be synthesized, which could ultimately lead to the toxicity, because more protein will lead to the toxicity. So in that case, this copy number alterations is mostly leads to diseases such as cancer, and we can detect this with comparative genomic hybridization methods or next generation sequencing. And these are very important because in cancer studies we mostly try to look for the copy number alterations if present. we can say that these genes, they get change in their copy numbers. And there are two kinds of genes. One is tumor suppressor genes and one are oncogenes. So oncogenes are the ones which are involved in the cancer, and tumor suppressor genes are the one which kind of try to suppress the tumor. So in such scenarios, the copy number alterations, they lead to the loss of tumor suppressor genes and gain of oncogenes. So these are two kinds of processes. And one other term which is very confusing is the copy number variants, and I have seen that many people use these terms interchangeably, that copy number alteration and copy number variants, but these two terms are not same, they are quite different. When we talk about copy number alterations, these are the alterations which are leading to change in the number of genes, and these are always leading to some kind of disease. These are the genomic aberrations which are involved in cancer. But when we talk about copy number variants, these are also present in the normal cells. So these are not linked to the disease. They can be present in the normal cells and they are present in the general population. And if you look at the impact of copy number alterations, that it involves the somatic events and relevant to identifying the driver somatic events in cancer. Whereas if we talk about copy number variations, so these are the germline variations. Germline variations, that means they can be inherited by the daughter from their parents or by the son from their parents. So these are the germline variations. And if we look at the focus of the study, so the copy number alterations, they are necessary to accurately identify and separate in the cancer patients. Whereas in copy number variants, it is useful to distinguish between the somatic copy number alterations from the germline copy number variations. So when we are comparing the normal and the diseased patients, then we can look for those variations which are common among both. They will be the copy number variations, and the variations which are specific to the cancer patient, those will be the copy number alterations. So these are two different terms, and copy number alterations are somatic, which cannot be inherited. They are occurring in the one person somatically on the somatic cells, and which are not inherited. And then there are some analytical approaches which you can use to identify, such as HMM dosage. So we don't have to go into the detail. This is just to clarify the concept that what is the difference. Don't get confused among CNA and CNVs. But the number of the copies of the genes, the phenomena is same in both of them. Either the number of genes could get increased or they could get deleted, but the difference is the occurrence, that where they are observed. Now let's talk about the genotype, phenotype and gene expression. So when we say genotype, it is the genetic makeup of an organism, when we say which genes are present. But when we say phenotype, so phenotype refers to the observable traits which we see after the genotype being expressed. So phenotype is kind of visually means which can be visually seen, but the genotype can't be visually observed. So that is main thing about the genotype and phenotype. And in the left figure, if you see, so genotype, so biological mechanisms, direct activity of a protein or RNA, and the phenotype could be biochemical and physiological properties, reaction product of an enzyme, and phenotype could be morphology, developmental processes, behavior, products of a behavior. And in the right side, the right figure, if you see, so the DNA is a genotype, which means the genes which tell the body how to do things. Whereas if we talk about phenotype, so thumbprint is the phenotype, that means the body organizes based on what genes say. And similarly, genotype will be a DNA gene containing the DNA for the height. The phenotype will be the height which you observe. So which genomic combination is present, it will govern the phenotype. And between that, there will be the interactions, there will be epigenetic changes, there will be many other things which will be playing role, which will be determining that what kind of phenotype will be observed after the genotype. Let's talk about GWAS, which is the genome-wide association studies. So as the name suggests, genome-wide association study, that means if we are comparing the genome of multiple individuals and we are looking at what are the associations between their genomes, then it is known as genome-wide association studies. It is an approach which is used to identify the genomic variants that are statistically associated with the risk for a disease or a particular trait. So for example, if we are looking for the, for example, skin color, then what we can do is we can take the multiple individuals, multiple human individuals which are present in different areas, and then we can compare their genome and we can identify the genes which are responsible for the trait responsible for the color. So skin color, it is a polygenic trait in which multiple genes are playing the role. So in that case we use the genome wide association studies. We compare multiple genomes and then we can identify that this particular trait is linked to these genes. So those associations can be done with the genome wide association studies. Similarly, for disease prediction also we can do GWAS, in which what we can do is multiple cancer patients genome can be taken, multiple normal patient genome can be taken, and comparative analysis can be done to identify the genes which are more commonly observed in the patients and not that frequently observed in the normal people. So these all analysis comes under GWAS, and this method involves surveying the genome of many people looking for the genomic variants, and SNP is one among them, that SNP comparisons can be done. So in the left side I have given that SNP1, SNP2, SNP3. So there are multiple single nucleotide polymorphisms present in individuals. So we can identify those SNPs and we can compare between the normal and patient, and we can see that which of the SNPs are conserved or which SNPs are unique to the individual with the disease. So in that way those variants can be identified, and you can compare the difference among both the SNPs in disease state and non-disease state. Okay, now let's talk about QTL studies. So to study the interaction or association between the genotype and phenotype, we go for the QTL studies, which are the quantitative trait loci, and this is a statistical method which links two types of information, the phenotypic data and the genotypic data. So phenotypic data here is the, for example, a cancer patient or a normal patient, it is a phenotype which we observe, and then genotypes means their sequences, their genomes which we are comparing. So when we are kind of commenting something from its genotype towards the phenotype, it is referred to as the GWAS studies, and they can be studied with the help of quantitative trait locus statistical method. So it is the attempt to explain the genetic basis of variations in complex traits. So it could also be used for the traits as well as for the disease conditions, and QTL analysis allows the researchers to look for the regions in the so agriculture people can also study that which of the regions are involved with particular trait. For example, some crop is giving the higher yield, some crop is able to survive in the drastic desert like conditions, so it is kind of heat tolerant, or lesser water conditions also it can survive. So we can identify those sequences or those regions which are present in those crops. So in that case QTL studies are quite helpful. They help to quantitate that which loci is mostly associated with particular trait. So these all studies comes under QTL studies, and goal is to identify the action, interaction, number and precise location of the regions. So in disease we can look for SNPs. If evolution, we can talk about the regions which are conserved among multiple individuals. For example, if we look for the evolution among monkeys to human, so there are multiple species which are present in between, so we can see which regions are conserved and which region has changed, so that we can comment that these changes have led to the evolution of human. So how humans have evolved from the primitive organisms. So these all studies comes under QTL when we are taking multiple genomes to study multiple genes. And now we can integrate GWAS and QTL studies. So the QTL studies can be of multiple types depending on which data we are analyzing. So if we are analyzing methylation data and single nucleotide polymorphism, it is referred to as MQTL. If we are looking for the expression changes, that with the SNP, what is the effect on the expression, how the transcription is influenced, it is referred to as EQTL, which is the expression QTL. And GWAS means when we are checking that how this SNP, which is the genotype, is leading to change of a trait. So it is referred to as GWAS, that means genome wide association studies. Similarly, there are MQTLs, PQTLs, depending on what analysis you are doing. So in EQTL I have slightly elaborated. So previously, as I was telling you about cis EQTL and trans EQTL, so in the rightmost figures, if you see the top one, this is showing the EQTL. EQTL is the expression QTL. When we say cis EQTL, that means there are some sequences or some genes which are present before a gene and it is influencing its expression. This could be a promoter region which is lying nearby. So in cis-QTL, the region is lying close proximity to the gene and it is influencing its expression. When we are trying to identify those regions or those sequences which are influencing the expression of a gene, this is referred to as cis-EQTL, cis expression QTL. Similarly, if that particular region is influencing the another gene functionality or another gene expression, then it will be referred to as trans-EQTL. That means they are present on the different regions or on the different chromosomes or very far away. So that is referred to as trans-EQTL. And in the bottom figure I have provided you that what could be the difference to call something to be a cis-EQTL or trans-EQTL. So cis gene, when the distance is less than 1 MB. But if distance is more than 1 MB, that means more than 1 megabase pair are present, then it is referred to as transgene and it will be a trans-EQTL which we will perform. So this is all about the EQTL, and there are some papers you can go through to get more detail about the genotype-phenotype interactions. In one such paper, there were two hypotheses which they have proposed. One was that DNA variation is directly linked to the protein level interactions, that gene decides which protein will be formed, and the another hypothesis was that no, gene is not sufficient to govern which protein to synthesize, but there are other factors also which are influencing, such as the epigenome, transcriptome, and then ultimately proteome will be synthesized and that will govern the phenotype. So genotype itself cannot govern the phenotype directly, but there are other agents that are involved. So in this, it's all about that hypothesis, that how they have performed experiments and how they have predicted in the end that genotype itself is not sufficient to predict the phenotype. There are other agents also which we need to study. And there was another paper which also I have attached. In this also they have compared the SNPs and then they have taken the genome of multiple individuals and then they have studied the SNPs, compared among them, and ultimately they have told that how to perform the GWAS studies. So you can go through it if you want to study GWAS in more detail. And these are the statistical tests which can be performed. So first is chi-square test, which is performed between the two categorical variables, and the example is that, for example, you want to determine the association between the smoking status and the lung cancer. So in that case you perform the chi-square test, that how many people who are smoker or non-smoker, how many of them have the lung cancer or they don't have lung cancer. So in this case it is like for the categorical variables, the output will be yes, no, there will be no numbers. If you talk about the Fisher's exact test, so it is also between the two categorical variables, but it is for the small sample. So it is similar to the chi-square, but it is for the small sample size. In chi-square you require the larger sample size. So in Fisher you require smaller sample, and for example you want to look for the relationship between the rare genetic mutation, that whether if this mutation is present, what are the chances of disease occurrence. So disease will occur or not occur, and this particular mutation is present or absent. So that is for the Fisher test. Then comes student t-test. It compares the mean of two groups. For example, if you want to check the average blood pressure levels between the two age groups, that what is the average blood pressure among the adults and the elderly people who are very old age people. So you can compare among them, young adults and elderly, and what is the average blood pressure. So it will be kind of number, it will be the mean blood pressure which will be given, so it will give you the number. Then the ultimate test comes, the Cochrane-Armitage test for the trends. So this test is used for the ordered data. So ordered data means, for example, you want to check the cell response if you increase the dosage of drug in the cancer cell lines. So because you are having the ordered data, you are increasing the drug in the ordered way and you are checking its influence, that how it is influencing the cancer cell lines, how the response is observed. So for that you will go for the Cochrane-Armitage test for trends. So these are few of the tests, because this was also involved in your assignment. So you can read them in detail because I have given the other key features also. So there are some MCQs. So I will quickly go through them because we don't have much time. Okay, so first MCQ was, Which of the following is the most frequent type of DNA polymorphism? First one is copy number alteration. Option B was single nucleotide polymorphism. Option C is deletion, and option D is insertion. So in this you have to tell that which of the DNA polymorphism is frequently observed. Okay, so anybody wants to answer? Okay, then I'll quickly tell you the answers also. So its answer is single nucleotide polymorphism. It is most frequently observed as compared to copy number alterations, because copy number alteration is linked to the disease such as cancer, so it can't be frequently observed. Deletions and insertions are also, they also come under the mutations which could be harmful for us, but single nucleotide polymorphisms, they are not that drastic effects, so they are most frequently observed among individuals. Then comes, which of the following statements are incorrect about the phenotype? Option A was it is produced by the interaction of the genotype and the environmental effect. Option B was it is observed trait of an organism. Option C was the binary trait is a measurable phenotype that depends on the cumulative actions of many genes and the environment. And option D was the quantitative trait is a measurable phenotype that depends on only two possible outcomes. So yeah, its correct answer is option number second, which is C and D. So why C and D is incorrect? Option A is correct because yeah, it is governed by genotype and environmental effect interactions. Yeah, it is observable trait which can be observed. But if you see option C, the binary trait. So when we say binary, binary is often mentioned for when there is two is involved, when the two outputs are involved, either 0, 1, yes, no, that is binary. So binary trait is a measurable phenotype which depends only on the two possible outcomes, which will give you only two possible outcomes, whereas if we talk about the quantitative traits, which means they are not governed by only two outcomes, there are cumulative action of many genes. So for example, skin color is also a quantitative trait. So in that way, C and D options were incorrect. Now let's look for the question number 3. Which of the model or test cannot be used for the genotype-phenotype association analysis in binary traits? So when we are only looking for the binary traits, then which of these tests could not be used? So its answer is quantitative trait loci, because as I mentioned, quantitative trait loci focuses on multiple genes, not only on the two genes. That's why QTL is the test which cannot be used, and I have also explained the other test. So Cochrane-Armitage trend test we already discussed. When we want to check, it is for the ordered data set, when we want to check the effect of the drug dosage on the patients. Recessive model test is also to test if the specific genotype is associated with the binary trait or not. Dominant is also used for the binary trait identification, and allelic associations is also used for the binary traits, whereas QTL is for the multiple gene analysis. And the last question is, in genomics, a genome-wide association study, also known as whole genome association study, is an observable study of a genome-wide set of genetic variants in different individuals to see if any variant is associated with a trait. Which of the following can least explain by the genome-wide association study? So first one is rare single nucleotide variants, de novo variants, copy number variants, and gene-gene interactions. So you have to tell that which of these will be least explained by the genome-wide association studies, and the correct answer is de novo variants. So if we talk about rare single nucleotide variants, so we can identify with the genome-wide association studies because we can compare the genome of multiple individuals and we can see that which of the SNVs are not majorly observed. So they will be rare single nucleotide variants. And if we talk about the copy number variants, they can also be identified. For the multiple genomes can be compared and copy number variants can be identified. Gene-gene interactions can also be identified. But if we talk about de novo variants, these are the variants which are generated suddenly, which are not inherited. So these variants could be difficult to detect by the genome-wide association studies. That's why this can be least explained. And in the end, I have provided you about the how to go through the brain base, how to explore it, and it will tell you about different cancers or different diseases which are associated with the brain. So you can follow this up in the slide. I have given you each and every step that what you can follow and how you can analyze the data in it.
So if you are interested, you can go through it. I won't explain it, and if you have any doubts, then in the next session you can ask me. So this is all for the session. I have taken 6 more minutes, sorry for that. So if you have any questions, okay. Yeah, so actually I have also going through your assignment, so I also come across sexy. So there is no such technique as sexy. There is a technique which is capture DNA capture sec, but sexy is not any technique which I have seen anywhere. So I cannot comment on it, because even if you search anywhere, you won't be able to find the sexy. So I'm confused. If they have written the capture sequencing, then also it comes under high C, but otherwise there is no such term sexy. It's quite confusing. Means they should have mentioned it, like, yeah, I cannot answer, but I can only say that there is no such technique which is sexy. I think they should have clarified it. They should have either given the DNA capture sequencing, because I think you should leave it, but I'm not sure that what could be the right answer. Because the confusing part is that in the some option they have also given that both high C and sexy, so that is quite confusing. The sexy is somehow kind of giving the hint that it is related to high C, but I can't find it anywhere. I have seen many papers. I don't know who have designed the assignment, so might be they have thought of something and then they have given sexy. Okay, so I will also look for the answers and then I'll see. Okay, so are there any doubts? Okay, if there are no such doubts, then I'll then end the session. Thank you so much for being here, and I will upload these videos, and I will also give the link to the slides in the description section also, and in the spreadsheet also, which you will find on the NPTEL. So thank you so much. Goodbye, good night.